Why plants making all these useless compounds?
Useless ah? If plants don't make this kind of products, insects will eat them as biryani. Do you understand, useless fellow? Plants produces two kinds of compounds. The first one we eat in tiffins, lunches, dinner. The second one we cannot eat it. It is for protection purpose. Now let's start today's topic. Plants are producing primary and secondary metabolites. The primary metabolites are carbohydrates, lipids, and proteins. So we are eating these primary metabolites. So now you must have wondered what are secondary metabolites. These secondary metabolites are used in defensive and interaction and adaptation purposes. So these are helpful in protecting the plants from the herbivores, pathogens, UV radiation, and also for useful for attracting pollinators. Secondary metabolites are broadly classified into three major groups based on their biosynthetic origin. They are terpenes, phenolic compounds, nitrogen-containing compounds. Now let's understand each category in detail, including their biosynthesis and their roles. The first one is terpenes. We also call it as isoprenoids.
Oh, these plants also have self-defensive systems?
Exactly. And these useless compounds are called secondary metabolites. Terpenes are the largest class of secondary metabolites. These are built with 5 carbon units. We call it as isoprene unit. So from this isoprene unit, all terpenoids are synthesized. So it is the precursor for every terpene molecule. How these terpenes are produced? Terpenes are synthesized through two pathways. They are, the first one is mevalanoic acid pathway. The second one is non-mevalanoic acid pathway. The first one occurs in the cytosol. It starts from the acetyl-CoA. 2-acetyl-CoA converted into 2-acetyl-CoA. Acetyl-CoA converted into mevalanoic acid. Next it converted into IPP, that is isopentenyl pyrophosphate. What is IPP? Do you know? IPP is precursor for terpenoids. So many of the terpenes are produced from the IPP. There is also another precursor, let us see it later. The second pathway is MEP pathway, that is non-mevalanoic acid pathway. Okay, it occurs in the plastids. It starts from pyruvate as well as glyceraldehyde 3-phosphate. These both pathways produces IPP as well as DMAPP, that means dimethyl allyl pyrophosphate. We call this IPP and DMAPP are universal building blocks for terpenes. There are terpene types based on the IPP combination. You can see that 2 units of IPP molecules produces monoterpenes, that is C10. Example for it is menthol. Do you know what is menthol? Please comment. 3 units of IPP combines and forms sesquiterpenes, that is C15. So 3 units are forming sesquiterpenes. 4 units are forming diterpenes. 6 units are forming triterpenes. 8 units are forming tetraterpenes. Tetraterpene means C40. You can see the examples as well. Artemisinin, gibberellins, steroids, carotenoids. Okay, remember them all. What are the functions of the terpenes? Let's see. They are useful for attracting the pollinators because they produce a kind of scent. They act as a defensive compounds to kill the insects. Some terpenes function as plant hormones like gibberellins. So they may useful for cell elongation. Carotenoids protects plants from photooxidative damage. Did you remember we have studied it in previous classes? These are important for economical uses like essential oils and rubber.
Is the plants producing smell for enjoying purpose?
No. Do you think plants are running perfume shop for you? They are for their defensive system, not for you. They are also useful for attracting the pollinators and also to warn the insects. Phenolic compounds. Phenolic compounds are characterized by the presence of aromatic ring with hydroxyl groups. Biosynthesis of phenolic compounds. Phenolic compounds are synthesized through shikimic acid pathway, which is occurring in the chloroplast. The starting precursor molecules are phosphoenol pyruvate, erythrose 4-phosphate. Where PEP is produced? Did you remember glycolysis? So it is produced there. So phosphoenol pyruvate plus erythrose 4-phosphate produces shikimic acid. Now it is converted into chloricimate. Now it is converted into phenylalanine. The enzyme phenylalanine ammonia lyase, that is PAL, converts the phenylalanine into cinnamic acid, which is the starting point for many phenolic compounds. Remember one thing, chlorishimate is the central intermediate molecule for phenolic biosynthesis. So it is a central molecule. Remember this point, okay? Types of phenolic compounds. Phenolic compounds include flavonoids. Their function is for pigmentation. Pigmentation means forming colors. So here they are useful for flower colors. The second phenolic compound is tannins. These are helpful for defensive system. The third phenolic compound is lignin. It is useful for structural compound. Lignin is a hard compound, so it is helpful for the plant to stand. The functions of phenolic acids are they protect against UV light. They provide mechanical strength. I told you just now. They act as antioxidants. Antioxidants are helpful for reproducing the dead cells. They are useful against pathogens. Okay. The third one is nitrogenous secondary metabolites. These compounds contain nitrogen and they are derived from the amino acids. Biosynthesis. These amino acids converted into nitrogenous secondary metabolites. The amino acids are tryptophan, tyrosine, lysine, ornithine. So these amino acids are converted through decarboxylation and modification reactions. Major groups, alkaloids.
Sir, why these alkaloids are toxic to us?
Imagine you are a plant.
Okay.
Cows comes to eat you.
Okay.
You cannot run.
Okay.
Cow started eating you.
Okay.
So how you escape from it?
I don't know, sir.
Do you understand now? So these compounds are helpful for protection of plants. The cattle don't want to eat again if they eat one time. Alkaloids are basic nitrogen containing compounds. Examples of alkaloids are morphine, nicotine, quinine, caffeine. Now you understand what are alkaloids. These are very dangerous to us. I already told you one thing that these are produced from amino acids undergoing some modifications. The second defensive compound is cyanogenic glycosides. These compounds releases hydrogen cyanide. You already know what is cyanide, okay? It is a very dangerous product. It is produced when plant's tissue get damaged. It is not that powerful as real cyanide, okay? But this can stop the oxygen usage in the lungs. So digestion and respiration will be hard for the animals. The third compound is glucosinolates. It is present in the mustard tree. It releases mustard oil when damaged. Roles. What are the functions of nitrogenous secondary metabolites? Strong defensive system against herbivores. Toxicity helps in plants protection. Many have medical importance. So these are useful in medical field like morphine. And they are also involved in the allelopathy. That means inhibiting other plants. What is allelopathy? Some plants prevents the other plants growth by releasing some chemicals. So we call it as allelopathy. Like we do black magic on the neighbors if they develop. Integrated understanding. So now we can clearly see that secondary metabolites are not waste products. These are specialized compounds that can help the plants to survive and interact with the environment. Let's see again. Terpenes built from the isoprene units. Phenolics derived from the shikimic acid pathway. Nitrogenous compounds are derived from the amino acids. So each group has specific biosynthesis and as well as ecological function. There is a way how to remember these all. How to remember terpenes? These are produced from the acetyl-CoA, which is a energy-based molecule. We know where it is produces. It is produced after the glycolysis before the Krebs cycle. The second one, phenolics. It is produced from the sugars. It is useful for protective compounds. The third one is nitrogenous compounds. It is produced from the amino acids, useful for toxic defense. I am also showing you some important compounds. Please remember them all. These are important, so please remember this. That's it guys, we have completed secondary metabolites. Now there is only one topic. Please wait for last topic of plant physiology.